Good evening, everyone. My name is Senthil, as Chris just introduced. I'm the founder CEO for ONS Bio LLC, which is a Cleveland, Ohio-based company, life science company, and also the Midwest territory distribution partner for ROKIT Healthcare. It's my great pleasure to talk to you today. I thank once again Dr. Dae-Seok Yom and Bogie Anderson and you all for the opportunity and time today. I will now turn off the video just to make sure there is no video interruptions or poor connections. All right, thank you again. So today I'm going to be talking to you about the bioprinting as a powerful research tool and a potential therapeutic platform in the context of emerging megatrends in science and healthcare. I understand from Dr. Yom and Anderson that most of the attendees today are skin researchers. So I have tried to customize the presentation to focus more on the bioprinting of human skin equivalent and related applications of the skin models. I will also touch upon other areas of application towards the end, so you get an idea of the wide breadth of applications that is relevant for bioprinting across many different disciplines. In addition, beyond the basics of bioprinting, which I'll introduce, I will try to cover some exemplary real-life applications and case studies of the ROKIT's 4D bioprinting technologies from research labs, commercial industries around the world, and even the operating room. ROKIT Healthcare is a South Korean pioneer and leader in regenerative medicine. Notably, ROKIT Healthcare is the manufacturer of 4D bioprinters and organ regeneration platform technologies, namely INVIVO 4D, Dr.INVIVO 4D2, and 4D6, with, which have revolutionary potential and applications in both basic and translational research, and also in the development of autologous therapies. So it's worth noting that in addition to the bioprinters mentioned above that ROKIT manufactures, they also manufacture industrial 3D printers and also offer genomics and single cell RNA services among the other things that they do. So today's talk is part of an active outreach to researchers such as yourselves across the US to educate and create awareness about the potential for bioprinting technologies to transform and advance your research. This is done as a partnership effort between ROKIT Healthcare, my company ONS Bio, and Indicate Technologies, based out of Santa Clara, California, in its capacity as the sole distribution partner for US West Coast customers, such as the UC Irvine. So I'm joined by my colleagues, Chris Kamimura and Parker Bovenberg, both from Indicate Technologies, as well as Dae Lee from ROKIT Healthcare. She will be joining us, or she's already joining us from Seoul, South Korea. And together we make up a team of bioprinting and 3D printing specialists that you can always refer to for any on-demand support anytime. All right, without much wastage of time, I will move on to the next slides. So what is the driving need or driving force behind the use of bioprinting of tissues such as human skin equivalent? So there are several megatrends in biomedicine that give us reasons to pursue and innovate in the area of bioprinting and engineering of human tissue models. For example, in 2018, we saw the feature news of a boy who was living for 14 years with a new bladder made from his own cells, seeded with 3D inkjet machines into decellularized pig bladder as a scaffold. This was a work done by the renowned tissue engineer and surgeon, Professor Anthony Atala, that many of you may already know. In July 2019, the American Medical Association approved reimbursement codes for 3D printed surgical models and tools, making the foundation for a clinical registry of patient 3D scan data and relevant 3D modeling, which will serve as learning resources and tutorials, and also provide the much needed impetus for active implementation of 3D printing in biomedicine. In September 2019, the United States Environmental Protection Agency announced that it will eliminate all mammal testing by 2035, becoming the first federal agency to make a strong statement and commitment to the transition to humane research, while emphasizing the urgency to find alternative solutions to the use of animals in research. And in 2019, ROKIT Healthcare and the Diabetic Foot Ulcer Clinic in India put the bioprinter, Dr.INVIVO, in the operating room to create a streamlined process of skin regeneration for diabetic foot ulcers. This new and alternative method for chronic wound healing promotes skin reconstruction using bioinks made from patients' own adipose tissue, printed into size-specific patches and applied directly onto the wounds. These dermal patches integrate with the skin, and through early onset healing, which is kind of the ROKIT's mission, part of the ROKIT's mission to decrease the rate of amputation for patients with DFU, diabetic foot ulcer. This is critical, especially given the fact that the 5-year survival rate of patients who have undergone amputations is lower than the 5-year survival rate of many cancer patients. Now, in the context of a need, in the context of need for human skin equivalents and models that are created either through bioprinting or other methods, 3 trends are notable. Namely, animal testing bans in cosmetic and pharmaceutical industry is becoming very widespread, both nationally as well as internationally. Second is the dire need for better human physiology and disease models, which has come with the understanding that animal models and 2D skin models simply do not recapitulate what actually happens in the human body. So many studies have been done that demonstrate chemicals which are non-irritants in rabbits actually end up being irritants in human skin. And also studies, especially in cancer, have shown that 2D cell cultures are more resistant to a therapy than 3D cell cultures, which leads to skewed decision-making in the evaluation of novel therapies. And lastly, the other important trend is that all of this leads to the third reason that is driving the need for human skin equivalents and other bioprinted solutions. That is the need to save time and the costs of research and development. The R&D costs are going up each year, but ironically, the health budget of each country is becoming more strained. The only way to resolve this would be to create a fully human cell-based testing method so that we don't have to go through as much animal tests and also have failed clinical trials as we've been seeing so far. So one concept that you may benefit knowing and might appreciate is the principle of 3 R's, which is the replacement, reduction, and refinement. So this is a concept that was developed 50 years ago, providing a framework for performing more humane animal research. Since then, it has been embedded in both national and international legislation and regulations on the use of animals in scientific procedures, as well as in the policies of organizations that fund or conduct animal research. So the background that I'm trying to set here basically underpins the fact that ethical testing is no longer a lifestyle choice. Rather, it is soon to become a legal requirement in the processing and manufacturing of new products, because the 3R that we just saw calls for models that are more sustainable, more biologically efficacious, and at the end, more cost-effective to the biomedical ecosystems of today. And last but not the least, in the most recent months of 2020, COVID-19 has changed everything about the way we live. And most notably, we have seen that the pandemic disrupts the traditional centralized value chain in manufacturing. For example, when the hospitals got cut off from supplies of masks and ventilators because factories were shut down or they were not accessible, 3D printing, additive manufacturing came to the rescue to manufacture these supplies on demand and on site. So if we put all of this together, all these mega trends that we are observing together, it points to the fact that 3D bioprinting and 3D printing or any facet of additive manufacturing can react directly to decentralized demands from life sciences for life sciences research and as an effective medical strategy. So clearly there is a growing value for an on-demand biomanufacturing, and bioprinting is quite relevant to the real world needs, both in research and the clinic, in keeping up with these observed megatrends. So in this half of the presentation, we will talk about what exactly is bioprinting and what some of its applications are. Just to give a quick primer for people who are new to this, here is a sample video of bioprinting in action, producing three-dimensional vascular structures with collagen inside a gelatin bath. Though bioprinting is getting a lot of attention in mainstream research only recently, the origins of the technology actually go back 2 decades. It's not totally in its infancy as many people may think. In fact, the number of publications around this technology have been growing exponentially since 2000, as you can see from this chart here. And today there are thousands of publications testifying to the benefits of this technology across tissue engineering, biomaterial development, regenerative medicine, and many, many other scientific disciplines. So here you can see a list of publications that are hyperlinked. There is already been a lot of evidence about the ideal choice and combination of cells and biomaterials for tissue-specific applications involving bioprinting. Perhaps in the near future, it is quite conceivable that journal publications will require data from bioprinted 3D human tissues as a go-between the 2D in vitro cultures and in vivo animals or human studies. And it may actually turn out to be an important criterion for your research to move forward. So what is bioprinting? Is it 3D printing or is it 4D printing? Well, bioprinting is an additive manufacturing process in which cells, biopolymers, and growth factors are combined to create tissue-like structures that imitate native human tissues. Because these printed living tissues are often further incubated to have them to proliferate, mature, and change over time, there is a temporal dimension that needs to be taken into consideration here. And so that makes the 3D printing that we popularly use, it takes it and converts it into a 4D printing. And in many ways, it's appropriate to call it 4D bioprinting rather than 3D printing, 3D bioprinting. So the 4D bioprinting technology basically consists of 3 components as shown here: biomaterials, the bioprinter, which is the hardware component, and then the design, which is the software component. So first, let's quickly talk about the biomaterials. Unlike the traditional 3D printer, which uses hard plastics, metal, and synthetic materials to create non-living structures, a 4D bioprinter uses materials that are biocompatible, biodegradable, and derived both from natural and synthetic sources. Naturally derived biomaterials, for example, include collagen, gelatin, chitin, coming from extracellular matrix of human or animal cells. Then they can also be alginate, sericin, or cellulose type material coming from seaweed, insects, plants, respectively. Natural hydroxyapatite comes from mammalian bone, and so on and so forth. So the synthetic biomaterials are biodegradable polymers with enough strength and elasticity to mimic human tissues and some of these have actually been used in FDA-approved medical devices. So they do have some real-world use cases. Finally, the composites may combine natural and synthetic materials to either add or enhance certain mechanical or chemical properties. Now, these biomaterials can be soft, low viscosity hydrogels or hard biodegradable plastics. And when hydrogels are combined with cells for printing, they are called bioinks. Bioinks basically serve as the extracellular matrix when you're trying to print cells outside the body. And so they, in that sense, they offer protection from shear stress, and they also provide nutrients and hydration and give the overall physiochemical support for the structure. So when selecting biomaterials or materials for bioinks, there are several key criteria which can be basically summarized into two broad categories. One is the bioinks should be able to extrude out of the nozzle tips, stack layer by layer structurally. Two, the bioink should be compatible and uphold high cell viability. So the factors such as viscosity, shear thinning, viscoelasticity or hydration degree, and its gelatin kinetics, all of these are important, and those have to be taken into consideration when picking something. Not to mention the biocompatibility as well. So now let's move on to the hardware component. So one key component of the bioprinting hardware, and I'm sorry, now that we have looked at the biomaterials, let's look at the hardware component. There are various methods of bioprinting that are available today, starting from inkjet-based methods to laser-assisted and the stereolithography methods. But among all of these, the extrusion is the most common one. And if I have to put a number, I would say around 70% of the bioprinting is done through the extrusion method. And that's what we are going to be focusing today. One key component of the bioprinter hardware is the print heads themselves. And they should be able to print both hard as well as soft materials. And depending on this, there are two ways that the print heads can print. One is in a scaffold-laden way, where they use hard scaffold materials like PCL. The other is a scaffold-free hydrogel that can be used to print. Now, another key component of any bioprinter is the chamber's ability to mimic a biosafety cabinet, because after all, you're dealing with living cells. So to optimize cell viability and functionality under the stress of printing, the chamber of a bioprinter should have UV disinfection, HEPA filter, and it should incorporate some bioreactor features and modules such as a carbon dioxide incubator or a temperature and humidity control, so as to provide optimal environment for the cells. Now, lastly, the third component of bioprinting is the design, meaning the 3D models of the objects we want to print. And that is the mostly the software component. So the 3D model files, which are known as STL files, can be obtained either by manual creation using design software, or they can be derived from CAD or MRI scans of the human body or tissues. These STL files then get processed with a slicing software into standard machine language that describes how a 3D printer should in a given three-dimensional space. And it includes information on the XYZ position coordinates, printing speeds, infill patterns, etc. The example here in the bottom are images below showing the difference between the STL object on the left and how the G code variations showing various patterns would be. Okay, now that we understand the components of the bioprinting, we can take a look at the bioprinting process workflow. The process principally involves 5 key steps: preparation, which involves designing the experiment with a clear identification of the problem to address. It includes selection of the right biomaterials, cell types, 3D models that the printer will use. For example, say you're printing a skin, then you will need to know and understand the structure, the layering, the different extracellular matrix components in each layer, different types of materials that would actually match the properties of the skin in the different layers of the skin, and what cellular compositions are. So those kind of background information all goes into the preparation stage. And then we have the printing, which is the actual printing process. 3D models are sliced into appropriate printing instructions by a software. Bio ink is placed in a printer cartridge, and deposition takes place based on a digital model. Now, post-printing is the mechanical and chemical stimulation of printed parts so as to create stable and functional structures of the biological material. Evaluation is the step in which the cultured printed tissues are assessed for structural or morphological and functional characterization in comparison with a living tissue standard. Application involves making use of the bioprinted material or print or product for various purposes such as disease modeling, in vitro safety, efficacy testing, and in vivo tissue transplantation, and so on. So if we take a look at an example of this process in the context of, say, cardiac patch engineering, it would look something like this. So in the interest of time, I will skip this so you can review this later when the recording of this talk is provided to you. As it is pretty intuitive. So bioprinting has been used in various research areas across tissue engineering, material development, regenerative medicine, veterinary medicine, toxicology, pharmaceutical sciences, and so on. It's a very long list. But if I have to summarize the applications of bioprinted products into 3 key areas, then they are in vitro efficacy test models for toxicological testing, screening of novel products, whether they are drug, cosmetic, or food. Number 2, the in vitro study model for human tissue physiology and disease modeling. Lastly, in vivo model for transplantation and regeneration. So these are the 3 broad categories of applications. Now, in the context of all of these, ROKIT Healthcare as a company is focusing on an organ regeneration platform that and has been contributing to the advancement of breakthrough science and healthcare through these bioprinting efforts and innovations. So ROKIT has dedicated dedicated a lot of resource and efforts into providing these offerings to over 25 countries around the world, and the user base spans across 250 different institutions and partners. And ROKIT has also been a pioneer and leader in introducing state-of-the-art bioprinters such as the Dr.INVIVO 4D, which when launched in 2016 was the world's first clean chamber bioprinter with integrated HEPA and UV lamps and hybrid print heads with the ability to print both soft tissue as well as hard tissue. And more recently, this year, they came up with the Dr.INVIVO 46, about which I'll be talking later in more detail, was to completely, the 46 was the first printer to completely integrate cell incubators and plasma sterilizers with 6 print heads for biofabrication to take tissue engineering to the next level. So in addition to the bioprinter and organ regeneration platforms, ROKIT also provides the other necessary components like biomaterials and the much needed education and training and support, technical support. So here, as you can see, there is a whole bunch of publications out there, many different case studies, and lots of users of the bioprinting platform from ROKIT. So I will get to the actual heart of our talk today, which is the applications aspect, which probably is what you're most interested in. So the following slides are actually going to be summaries of publications by Dr.INVIVO 4D users, unless otherwise specified. For more details on the journal publications that may draw your attention, any of these, just let us know later on. So we will guide you to them after this presentation. But my goal is to, since time is very short, my goal is to just quickly give you an overview and touch upon some of the interesting ones, and that way you can appreciate the potential and the diversity of applications of the bioprinting technology, as well as the use cases for Dr.INVIVO platforms. So to talk about skin applications, we have to know a little more about the skin structure. So the engineered tissue is, as you can see in this picture, the human skin equivalent is an engineered tissue that is designed to structurally and functionally mimic the real human skin. As you see in the diagrams and the H&D pictures above, a human skin equivalent, HSE, begins foremost with the structural simulation of the stratification all the way from dermis to epidermis. Now, the history of the HSC development goes back to the 1940s, when the culturing of 3D human skin biopsies as ex vivo explants began. But subsequently, it got, there was a lot of activity in terms of developing mixed and single type primary 2D cell cultures, including that of human keratinocytes, that added to this growth of this HSC development, to the point that around the 1950s, the relevance of 3D constructs became more evident. And in the late 1970s, a living skin equivalent implementing a dermis like collagen hydrogel was already described, with a reliable culturing method for human keratinocyte also established by that time. And later in the 80s and 90s, specifically, techniques like electrospinning led to the use of scaffolds for 3D cell culture. And moreover, at present in the 21st century, we see the implementation of 3D printing, 3D bioprinting for use in biological applications is replacing the hand-poured hydrogel matrix. So it imparts a level of automation to this process, and it's starting to set a new standard for production of architecturally complex 3D tissue constructs So besides all the scientific push that happened in the past decades, there has also been a great amount of regulatory push in the development of the human skin equivalent over the past 50 years. So in the early 2000s, we have seen the rise of many industry coalitions and national and international regulatory agencies pushing forward to make alternatives for animal testing. And so some of the examples of these organizations listed here are the global OECD, then we've got the European Society for Alternatives to Animal Testing, then we've got a bunch of other agencies. Now, these agencies and centers have been creating communities of researchers and regulatory scientists that are committed to publicizing and standardizing human tissues as animal alternatives in research and medicine. Now, this underpins the fact that ethical testing is no longer a lifestyle choice, but again, a legal requirement in the processing and manufacturing of new products, which I already kind of mentioned. So a key thing to note is that the OECD guidelines for testing of chemicals, which have provided in its portfolio as kind of a standardized way to assess efficacy of novel in vitro models for the potential effects of chemicals on human health and environment. For example, the OECD guideline TG 431 or 439, it offers standard ways of assessing chemicals that can cause skin corrosion or irritation. And these guides are continuously expanded, and they're also updated to ensure that they reflect the state-of-the-art science and techniques to meet all the member countries' regulatory needs. So the bottom line is the use of in vitro model is getting into the mainstream more and more. So to understand how to engineer a human tissue, we must first understand how the skin is structured. But thankfully, I don't have to spend too much time on this and this set of next few slides, because you all, as experts in skin anatomy and physiology, know way more than I do. So I'll just point out that there is a lot of layering here, a lot of different cell types here that involve a lot of different physicochemical properties and biological properties and immunological aspects to keep in mind as you are designing the skin or the human skin equivalent. So I will quickly glance through these because we don't have to dwell on these biology aspects, and we can go straight to the application aspects. All right, so here, this is a slide to quickly drive home the point of the limitations of animal and 2D models. So traditionally, testing of compounds has depended on animal tests and 2D cell culture methods. However, various research studies over the years have demonstrated that, that the lack of these models to fully recapitulate what happens in actual skin is a real problem. So if you take a look on the left-hand side, this is data on the patch test done in rabbit models. Without going into the detail, the take-home message here is that the animal models, they always or more often lead to incorrect labeling of irritants. Now, on the other hand, on the right side, you see a comparison of 2D cell models to 3D cell cultures in the context of testing survivability of the cells against cytotoxic agents. So as you can see in the graphs, to the same given dose of hydrogen peroxide or silver nitrate, the 3D cell cultures are twice as likely to survive as the 2D. So in addition, the study has shown that the cells in co-culture were better at surviving these stresses than in monoculture, which brings home the necessity for building increasingly more complex tissue for better modeling than relying on monocultures. So against the limitations of the animal models and existing 2D models, various three-dimensional fabrication methods for human skin have been developed. I'll quickly go through the most common ones. The most common ones include the freeze drying technique, where which is mostly a technique for scaffold preparation. It relies on the sublimation process in which frozen water in the polymer nanocomposites is directly solid to a gas state and then removed. Then we have the solvent casting salt leaching method, where addition of solvents to a polymer solution and hardening, followed by leaching of salts by submerging it into a solution that dissolves the salts away. Then we have the electrospinning, where the creation of nanofiber mesh applying very high voltage electrostatic forces to combine biocompatible polymers. And then we've got the decellularized pig skin models, where the combination of human cells to decellularized pig skin to serve as an extracellular matrix scaffold. Then we've got the spheroids, which involve the seeding and growth of micro-sized cellular aggregates. While a lot of these three-dimensional fabrication methods are a large step forward from existing animal models and 2D human models, they are not without limitations themselves. For example, they have difficulties in controlling cellular composition and deposition at a macrostructure level. Then there is the adverse effect of pre-processing, data inconsistency due to human error, and also limitations of applicability downstream because of animal-derived components. Then we also have problems of necrotic cores because it's very hard to reproduce vasculature. So if you see in the picture in the bottom, you will see that on the right is human skin equivalent made by using 3D bioprinting approach. And then that's compared to the, the manual casting approach. So while the bioprinted one obtained a perfectly circular shape through an even distribution of substrates, the manual casting approach led to a disfigured shape that ultimately resulted in abnormal pigmentation in the center. So here is a sample workflow that is of what the human skin equivalent preparation process might look like. And these are actual images taken from the ROKIT Healthcare's skin R&D team during their Epitem product line, which is a human skin model that they offer. As you can see, the stratified layers of Epitem human skin equivalent from ROKIT are realized onto well-played formats. And this allows the use of these human models for adopting them into high throughput assays, say like drug screening. All right, the applications of human skin equivalent. Now, what are the applications of HSE today that we know of? The applications are numerous, but they may be divided largely into three types. One is the in vitro efficacy test models for toxicological testing and evaluation of novel drug or topical agents. Second is in vitro models for skin disease modeling. That is, and then lastly, we have got the in vivo models for transplantation and skin regeneration. First, let's talk about the in vitro efficacy test applications. Quickly, the corrosion and irritation tests with the HSE, they've already been established as per the OECD guidelines titled TG 431 and 439, and these allow for standardized evaluations of alternative methods for animal testing. A substance, for example, is applied to the surface of the HSE to check for toxicity within 4 hours. And then we've got the absorption permeability tests that are possible, and that can be done by measuring the amount of transdermal moisture loss in the human skin equivalent, or through immunostaining of the moisture-related proteins. Then we've got the sensitization test, that can be used for experiments related to immune system, such as hypersensitivity to certain substances, as in the case of erythema. This can be done by exposing the allergens to the surface of the HSC and then identifying the immune reaction mechanisms. Now, with an HSC made by mixing melanocytes and keratinocytes, you can perform lightening or whitening tests, which tracks any changes in melanocytes in response to a substance entry. The HSC also finds application in the pharmaceutical industry, from novel drug screening to drug delivery mechanism studies. It's also possible to judge the presence or absence of toxicity with the aid of phototoxic substances. It is also possible to check the degree of aging of the skin by conducting a UV transmission test. And in case of the aging skin model, it can actually be created by manipulating the dermis layer. The effectiveness and toxicity of anti-aging related cosmetics may be evaluated that way. And with the use of the COMET assay, it is even possible to check the presence or absence of genetic toxicity by analyzing DNA damage in the HSC. So as such, there are numerous efficacy tests already done with the human skin equivalents. Compared to the conventional single cell test methods, the HSC allows you to obtain more accurate data in a more human-like skin environment. And today, the range of applications of HSC is only growing, so making it a versatile alternative for animal testing. Beyond the toxicological efficacy studies and tests, the HSC is today widely used as a disease model as well. Skin cancer is a common cancer worldwide, as we all know, with a range of types, including the basal cell carcinoma or squamous cell carcinoma and melanoma. We can imagine the HSC made from a mixture of cancer cells and skin cells can help study cancer-related gene pathways, the expression profile of the tumor suppressor genes or genetic mutations, or any damage to the tissues occurring from cancer. So the wound healing assay, which is a representative disease model of animal experiments, can also be performed using the human skin equivalent. For example, first, a model of the chronic skin wounds is developed by shining a laser on a regular human skin equivalent. Then, over a period of time, say, for example, 72 hours, like shown in this picture, we can assess the wound closure process and mechanisms in response to a certain topical agent or a cellular therapy. It's also possible to study psoriasis and atopy, a representative autoimmune disease among skin diseases. Immune disease models can be induced by introducing various combinations of cytokines in the culture. In the case of psoriasis model, we observe irregular epidermal, in this picture as you see, we observe an irregular epidermal structure with a thinner than normal area. And the atopic model, on the other hand, forms a sponge-like basal layer with a thinner than normal epidermal structure. So in these models where the expression of specific protein markers is reduced, various tests to assess pathological effects of drugs and gene therapy, their therapeutic effects and unique biological characteristics, all of these can be performed. So as such, the HSC offers a very attractive disease model that allows one to perform molecular biological analysis in a human cell environment that is much more clinically relevant than any traditional animal models. Other than the disease models, depending on which cell types you choose, the HSC can further open research avenues in skin physiology, in the exploration of skin physiology. We all know that the donor cells from infants and teens to 60-year-olds exhibit different epidermal structures. So this kind of shows that the skin structure and its behavior differs across age groups. So this implies that for a truly accurate skin research, there is a need for a model for each group. Now, you can also imagine creating models for hair regeneration using human skin equivalents, which would be of great interest worldwide. So a hair skin model would further expand our portfolio of tests to include tests for hair products or drugs related to the scalp, hair roots, or hair loss. So there are many possibilities here, and there are also other nuances to how the skin might be varying in color between races and so on, which I mean, which is essentially, I will skip this slide, but essentially driving home the point that there is there is a possibility for us to try out many different combinations at a much more cost-effective and realistic way. And in that context, the human skin equivalents or any bioprinting approach used to recreate the skin models is quite relevant. So quickly, here is some in-house data on the engineered skin tissue, EpiTem, available from ROKIT. The figure 1 shows the histological characterization of the engineered epidermis version. As we have seen before, the tissue shows all 4 distinctive sublayers of epidermis from stratum basale to the corneum at 18 days of reconstruction. And figure 2 shows the immunohistochemistry results, staining for markers like cytokeratin 10 and P63. And then besides the structural features, the engineered epidermis also has been tested against the OECD guidelines to assess functionality as an in vitro skin model. And in this case, we find the barrier function test and irritation test all performed to the expected standards. Another study that's not directly skin engineering related, but is relevant to is the engineering of adipose tissue using the bioprinting approach. Now, in this study by Seoul National University, a research group sought out to engineer a novel way to regenerate adipose tissues. Conventional medical grade materials like PCL were used. I mean, they are normally used in breast cancer, breast reconstruction and nose reconstruction. But the problem is these do not match mechanical property of the adipose tissue. So what this group did is they first developed a novel biomaterial called PLCL and mixed it with adipose-derived ECM. And using the bioprinting technology, the group created a size-customized scaffold, which when implanted onto mice, promoted adipose tissue regeneration with macrophage filtration and vascularization. So the group confirms that bioprinting combined with the new material that they developed that serves as the ECM complex, it offers a new solution for burn, trauma injury, or post-mastectomy repair, especially in large defects. All right, so we have looked at a bunch of applications that are more in the lab or in the bench side. Now, the most noteworthy and remarkable applications are always, or exciting applications are going to be when the applications are moving from off the bench into the clinic. So, thankfully, due to ROKIT's efforts, we have ROKIT has been leading an effort to reduce amputations in diabetic foot ulcer patients by offering a regenerative solution, one that leverages the patient's own autologous cells, tissue material with bioprinting technologies. So the result is an in-clinic bedside solution, and patients' autologous solutions, in this case, the adipose tissue-derived ECM, are extracted, printed, and placed onto the wounds in short time. This is patient-specific, site-specific treatment with no immune rejection. And based on a sample of ROKIT Healthcare's clinical study results in India and Korea, it took, as you can see in this slide, on an average of only 4 weeks to close the wounds that had otherwise stayed open for months to even years. The more global clinical studies are actually in line in the US, Turkey, and Italy in the upcoming year. And we envision that this therapy can be applied to more skin conditions, ranging from burns to scars to bed sores. So putting all this together, one can imagine that such a therapy can an integrated 3D scan print therapy, which takes the patient image, translates into a personalized regenerative solution at the bedside. And this video here will give you a rough idea of the workflow that was used in these studies. So basically, it starts with scanning the wound and the site of injury, and the software converts all the data into the digital files that are processed in the software engine, and subsequently we make measurements of the wound, and then that's followed by printing the scaffold, followed by printing of the bio ink from the patient's adipose tissue derived ECM. And that leads to the final step of actually placing it in the patient's wound in a customized fashion. It's very precise, personalized medicine. So far we have focused on the bioprinting of only skin, because I thought for this audience that might be of most relevance and interest. But as you all may very well know, bioprinting applications span numerous areas of research and tissue types. So I could easily spend a full lecture on each of these areas, but I will quickly go through some of these to give you a sampling of the diversity of applications before we can conclude the talk. So here in this slide is a study that was published in the International Journal of Molecular Sciences. What is being done is a 3D printed vascularized tumor model that can serve as an in vitro screening system for anti-cancer drugs. And the bioprinting approach provides the biomimetic tumor microenvironment. This is another example study. Now again, all of these studies, as you can see, have been done using the in vivo bioprinter from ROKIT. So here, this study, they developed an elastic nerve guidance conduit using a nanoporous structure with a material PLCL, which is polylactide co-caprolactone. And it combines with oriented 3D printed hydrogels, collagen hydrogels. And the conclusion or the result is that the aligned collagen hydrogels, they are enabled by, after a 3D printing process, the patterns that they generate, they provide a preferable environment for nerve regeneration, and that provides a guidance path along the longitudinal direction. So this method can be further enhanced by incorporating various other growth factors or bioactive molecules in the collagen as needed. And this is an application of bioprinting in the bone regeneration. So in this particular example, strontium was substituted or incorporated into the hydroxyapatite scaffolds. And the strontium incorporated hydroxyapatite bioceramic scaffolds were tested of for the optimal strontium content. And for the first time, they created a unique 3D print, a 3D model of the bone defect. And when incorporated and tested, the in vitro bioactivity test revealed that the strontium incorporated scaffold was able to better promote rapid cell proliferation, osteogenic differentiation, and cellular mineralization. So this really serves as a good novel bone substitute for in the context of large dimensional bone defects and autografts. This is an example of muscle model that was 3D bioprinted for drug testing using mouse muscle cells as an alternative to animal in vivo models. And this is an interesting example to drive home the point that the bioprinting approach or the 3D printing approach doesn't have to always involve cells alone. So here is an example of printing QR-coded drug delivery orodispersible film. Now, the long story short, what has been developed here is a QR-coded, a higher orodispersible film with a higher drug solubility. And it was successfully prepared, and it became useful in the context of enhancing patient compliance to prescribed drugs because of the QR coding. So this innovative drug delivery and patient info delivery device, it can enhance patient compliance to prescribed drugs and enhance drug solubility for patients with difficulties in swallowing. This is an example of an in vitro model for human pathological cardiac tissue via bioartificial scaffolds. So you can create predictive in vitro models for testing therapies, as well as myocardial regeneration after ischemic heart disease. Now, this is an example of being able to use the bioprinting approach to even the microbial applications, where in this case, inspired by the architecture of an eggshell membrane, the team developed a three-dimensional printing technique to realize bioresponsive devices with excellent selective permeability for effective biochemical conversion. And lastly, this is a case of, this is an application for printing a thermal therapeutic device, a wearable device. So the team successfully manufactured a wearable thermal therapeutic device from nanocellulose modified bio-based thermoplastic elastomer. And this technique can also be used to create a cancer cell-like environment for a drug screening platform. So the point I'm trying to convey here is that the applications are numerous across many different fields, because bioprinting is not a singular technology, it is a platform technology, and it is not restricted to any one discipline, cell type, or biomaterials. So the sky is the limit when it comes to its application, as long as the researcher is able to apply it in a creative fashion. So in the last few minutes, I will take you through a demonstration of some of ROKIT Healthcare's 4D printers quickly. As you can see here from this chart, the in vivo bioprinting platform users span many different institutions across the academia, industry, and hospital systems. And there are some notable names here in the US that you may quickly recognize, including the UCSD, or University of Illinois, Chicago, University of Michigan, Harvard Medical School, and so on. So when one of the important offerings that ROKIT has in terms of the bioprinter is the first bioprinter that they launched, which is the Dr.INVIVO 4D, which looks like this. It is a very powerful system that's capable of handling many different types of materials for use in many different applications, many different types of tissue engineering projects. And that includes use of bioinks, hydrogels with cell mixtures, or powder mixtures such as hydroxyapatite, hydrogels, or synthetic polymers like PCL, PLGA using the filament extruder, and pneumatic hot melt dispensers that can handle pellets, powders, and pastes, and so on. Now, ROKIT also is a company that listens to the users and the needs of the market, needs of the customers. So accordingly, after the launch of the INVIVO 4D, which I just showed, they did, and in line with their vision to take the innovations from the bench side to the bedside, they identified some urgent needs or unmet needs in the bioprinting space that would help and speed up the transfer of the potentials of bioprinting to the clinic. And so some of the needs were in the area of a need for in situ 4D printing. Why? Because the human body is a curved surface. So to be able to use a bioprinting approach for a direct application of biomaterial to a curved surface, such as the skin project that we described, it is important that the printer should be able to print in a curved fashion, in a Z-axis as well. Then the second thing is, if we want to take the bioprinter into the clinic, then it is important that it comes with very high standards for sterilization, because it has to treat all the substances as potentially infectious. Both the device should be able to sterilize itself as well as the implantable device that it creates. And lastly, to keep any cells that any patient cells or any cells that you use in the printing process must be always kept at the optimal condition to ensure maximal performance and survivability. So that calls for a bioreactor feature to be inbuilt into the design. And so putting all this together, ROKIT combined the Dr.INVIVO 4D capabilities with the incubation function and a sterilizer function to make it a well-rounded unit, which led to the INVIVO 4D6, which is a true organ regenerator with Z-axis curved printing technology, complete sterilization, including UV, HEPA filter, and hydrogen peroxide plasma sterilizer. Many of these features are not to be found in any other competitor bioprinter, just to mention. And an optimal incubation system that can maintain an optimal temperature environment and carbon dioxide level. So this is to give you a quick glance of how the printer moves or works. The notable thing here is it comes with 6 print heads on a rotary turret, and the 6 print heads and other capabilities of the bioprinter INVIVO 46 provide you the ability to recapitulate complex, intricate architectures, as you see in tissues like retina, bone, muscle, heart. All of that can be reproduced here using this. And other features such as the incubation, sterilization, and the bioreactor features, they provide the ability to control and provide the optimal conditions for the human cells and tissues during the printing as well as incubation phase. So in line with that, the printer comes with the plasma sterilizer, the HEPA filter, and also the temperature controllers, temperature and humidity as well, temperature, humidity, and carbon dioxide. And one other cool feature about the INVIVO 46 is that it also doubles up as a high throughput liquid handler, because when you are involved in projects for drug development or vaccine research or PCR genotyping, things like that, then you need a high throughput. And so the INVIVO 46 thankfully has the ability to handle even up to 384 well plate printing. Lastly, it comes with multiple print heads or hybrid technologies that let one use both soft, hard, synthetic, natural, all kinds of materials, and materials that may come into existence as the field matures and creates more innovations in that space. And it's important to mention here that as of now, the INVIVO 46 has the highest range of temperatures for the bed or the chamber or any of the print heads, along with pressure capabilities that you can't find anywhere else. So it is truly a remarkable bioprinter that will provide you the opportunity to apply for many novel applications. And also to mention, since one of the inherent motivation for ROKIT is to move the basic research into the clinic so that it's beneficial for mankind. The printer by design is CGMP ready, and then we do have a lot of hospital use cases. So it is for anybody serious about translational research and being able to develop products for the clinic, this is quite ready, good to go. This is a quick overview of the easy user interface of the bioprinter. It's quite intuitive and easy and simple. And the printer can be controlled from the computer or mobile device like a phone or a laptop or a tablet. Okay, now time for the last take-home message. So at the end of the day, bioprinting brings 3 breakthrough benefits to research and novel therapy developments. One is automation, physiological relevance, and on-demand fabrication. So what these breakthroughs result in? Ability to experiment with better repeatable and better physiologically relevant tissues. Ability to create biomimetic structures and tissues without having to depend on model systems that are too expensive or not ideal. The ability to create more sustainable research methods, phasing out animal models with alternative human cell-based methods of testing. And it's possible for us to imagine that bioprinting facilities would be set up in every region of the world, and the technology has the potential to completely transform regional biomanufacturing value chains, just as COVID-19 emphasized the need for the same. And it's not surprising, actually, to see that there are actually centers of excellence that have been arising at the national and international level over the last few years already. And here are some that you can follow. So lastly, we are entering an era of regenerative medicine. And in this era, bioprinting is not only a revolution that's underway for the way we create and use model systems for answering basic biological questions. It is actually revolutionary in the way that we will be practicing medicine. For example, instead of using the systems that we have relied on for so long to treat symptoms using small molecules or monotherapies, bioprinting as a technology is shifting the focus towards harnessing body's own healing potential that's inherent in our stem cells. So the future of medicine is already being realized today by these pioneering technologies and its application in various disciplines of science. There is a lot of possibilities and potentials, but it also comes with challenges, which I'm sure as a community, we all can take on and then make significant progress. So I really thank you all for your patience and attention. I guess I went a little past my 1 hour that I was planning for. I will be happy to take some questions now.
So what's the cost of these bioprinters?
Oh, thank you. So the cost of the INVIVO 4D, which is the first generation, is about $50,000 with lots of additional packages and all that. Now, when I say 50,000, I'm talking about, there are basically three different versions, and you can pick and choose depending on your intended application. One is the standard, the other one is upgrade, the other one is premium. And the cost I'm saying is for the premium version. Now, the cost of the upgrade and the standard versions kind of go down significantly. Say, for example, $40,000 and $30,000 for the other two. And then we've got the newly released INVIVO 4D6 with the 6 print heads. Now, the 6 print head and with a lot more automation and the other features that I mentioned in terms of the bioreaction and all that, that's somewhere in the price point of about $100,000. Now, all of that, when you compare in terms of features to any other competitor models, it is very well priced and packs in more features that might actually benefit diversity of applications. And needless to mention, all of this comes with ROKIT's full commitment, ROKIT and its partners like myself and Chris, our commitment to providing you the best-in-class training and support, and also continuous help in your in your experimental design and execution.
So, so the places that have these, let's say universities, like you mentioned UC San Diego as one. So what kind of entities within the universities are?
Oh, okay. At the UCSD, it's part of the bioengineering division, and at the University of Illinois Chicago, it's part of the orthopedic research center. It's both a clinical and basic science department. And then we also have it at the mechanical engineering department in University of Michigan at Dearborn. Actually, there are 2 units in University of Michigan, which I personally sold, so I know very well. The other one is part of the Biointerfaces Institute, where they do a lot of multidisciplinary research. Some people use for dental applications, many others use for other kinds of tissues as well. So if you are asking me if these are part of a core facility, the Biointerfaces Institute might be a core facility type of example because it's common for many different groups. The rest of them are mostly for individual labs.
I see.
Yeah, one could imagine a situation where you can pool up resource and invest so people can use for many different applications in a shared basis.
So they're actually being used already in the clinic. You showed a picture of one in an operating room. Is that so? They are already approved by to do that, or?
Okay, Diane.
Yep, I'm here.
Can you talk more about any potential FDA applications, things like that? Any updates there that might help?
Sure. Hey, Dr. Anderson, this is Daye, Senior Bio Consultant from ROKIT Healthcare. In response to your questions about the clinical product development using Dr.INVIVO, there are 3 key developments that are going on at ROKIT Healthcare's R&D center itself, together with some of our partners like the MGH in Boston, some diabetic foot ulcer clinics in India, and other various hospitals of cartilage regeneration divisions. And basically, there are 2 ideas I want to get across. One is there has been clinical product development using Dr.INVIVO in a GMP facility with approval for the process of the printing process of bone implants in a GMP facility. So the device is not just by the of the FDA for the process of printing standardized versions of bone implants in a GMP facility, and these implants are now being used in the hospital. But for the indications of diabetic foot ulcer, skin regeneration, or cartilage regeneration, we're taking a different approach. Instead of going through the long process of biologics or cell therapy approval by the FDA, we're actually going through what's called the in-hospital manufacturing process approval, which basically cuts down the long time that it takes for cell therapy, but goes through the minimal manipulation process clause of that process. So what that means is, basically, if we make use of minimally manipulated stem cells from the patient's own body, and if we do not culture them, but apply them directly to the wound beds in order to prime regeneration, we can get this entire process, including the device, approved for use in the clinic. So that's what has enabled us to already apply the bioprinting devices for in-hospital skin regeneration at DFU clinics in India, Korea, and now our team is now going to fly to the US next week for clinical studies in California with USC for DFU, and early next year in Turkey and Egypt. So long story short, again, either we take the approach of minimal manipulation for using the bioprinting device for autologous therapies, or we use the bioprinting device for printing FDA-approved implants.
So may I ask you questions?
Sure.
I guess my biggest question is, how does 3D bioprinted tissues are really, how similar it is to the human skin tissues? Yeah, I saw that it is structurally, you can make the layered structures, but in case of gene expression wise, how similar it is?
So, Santil, if you go back to some of the OECD data, I guess, so there is a guideline for testing by the OECD, the International Regulatory Agency, and they basically have a set of guidelines for specific tests, such as corrosion tests and irritation tests, that basically set the standards for the in vitro human tissue samples and how they compare with the human skin. And so these guidelines that they have set outlines some of the already known non-irritant chemicals and irritant chemicals, and see if the in vitro tests give you the same results as actual skin, then those particular samples would pass the guidelines for the use of in vitro testing with 3D printed human tissues. On the other hand, in the aspect of corrosion tests, they would basically have this set of guidelines for assessing how do the samples respond to certain cytotoxic agents, such as triton, 1% triton, and by assessing the loss of cell viability across time, they can test the burial function of the skin, and they can test the therefore some of the most important functional characteristics of that in vitro test sample versus human skin. So if we can follow these types of guidelines, these 3D printed tissues, 3D printed skin tissue models like the epidermis or the dermis models can be effectively used as standard ways of testing chemicals or compounds as for assessing how human skin may behave to these same chemicals.
So yeah, for us, we are studying the skin biology, right? So how the genes and proteins are interacting, how gene expression has been changed, something like that. So to be replaced by the real, human tissues, and we are most likely interested in the, at least there's some key, genes, expression of key genes can be recapitulated. So in the experimental tissue. So that's why I'm asking the questions. And then the other question is, what are the source of those cells you printed? Those are cells from human cell lines, or
these these are primary cells of neodermal fibroblasts, and they were cultured for 14 to 18 days until we see terminal differentiation of the keratinocytes into the corneum layer. And that's what you were able to see, the differentiation markers and the structural features as shown, the distinct microlayers of the epidermis model. And on the bottom, I think you might have seen like a whitish blue layer, but that's just the collagen layer that we print, that we develop this skin on in a air liquid interface of the well plate, just to make a clarification. And normally for these types of printed living tissues, we recommend that any sort of downstream applications are used within the 20-day mark since the initial culturing of these cells, because after the 20 days, based on our experiences and other publications, these living printed tissues continue to develop the corneum layer, where the keratinocytes from the below layers continue to differentiate, but then that causes the below layers to just collapse, meaning that your organized structure for the skin model begins to deteriorate, basically. So the longest time I have seen these tissues be used for is up to 30 days for certain things like long-term culture aging models, but most of the times it is used within 14 to 18 days.
I actually want to kind of follow up on that because the basal layer is totally flat, but in the human skin you have these papillae. We're talking about the two different types of dermis, the papilla dermis and the reticular dermis. So can you print it so that it has actually more like a more human-like epidermal structure with papillae?
Yeah, I mean, if you're asking about the ability to introduce certain cell types into the model.
Not cell types, but structure. So if you, I mean, early in you have a slide that shows like a picture of the normal epidermis somewhere. You can, this one. No, go back to that one. Next slide. This one, yeah. So that's like what normal human. The epidermis has these papillae, or has these structures that kind of butt into the dermis.
Right.
These ones.
Yeah.
Yep.
Can you? So when we do just the regular skin equivalent 3D culture, like
sure.
we use a devitalized dermis or any kind of artificial extracellular matrix, we get the thing. The junction between the dermis and epidermis is totally flat. But what my question is, could you bioprint it such that it forms these structures?
I see. In the existing, Yeah.
I'm sorry. My thinking is, so isn't that precisely where the curve printing comes into play, where we could print instead of a uniform layer, which most bioprinters do. This can go up and down to recreate those papillae or the cells in between.
Mhm.
Yeah. I think. But I think the papillary junction that exists between the epidermis and the sort of the dermis layers could be maybe recapitulated by just the use of, you know, the physical curve printing process of the skin. But most oftentimes, I think that part has more to do with the biology of things, the interactions between and the signal transduction between the two layers to cause that. So in our cases so far, we have not been able to recapitulate that that distinct, I guess, feature of skin. And I haven't seen publications from bioprinted or, you know, like man-made living human skin tissues that recap that. But I do know that there's a lot of research going on to sort of to maximize those characteristics, optimize those characteristics as well. And some have responded to this issue by saying that, you know, like some of the most commonly used cell types in reconstruction of these models are, of course, like fibroblasts and keratinocytes, but very, very little um, investigation has been done using other cell types that comprise the entire ECM and, you know, microenvironments of skin, like the immune cells and the whole ECM. And so if we go on to introduce these fine components into the skin, that would be important for recapitulating these fine details of skin as well. But more investigations would need to be done.
Mhm. Great. Well,
Plus,
thank
I don't
you.
know if I made it clear that today we talked about not only the printer and what and all you can do, maybe as an optimization for your needs. Maybe you can try to print the papilla. The epitome is a pre-printed, ready-to-go model of human skin equivalent. So those are two different products or offerings.
Mhm. So you guys are selling this epiterm.
Right, Dai?
Yeah,
So
well, so we sell both the already sort of ready-made skin tissues, the epidermis model. The dermis model is soon to come out next year. And we also provide for on-demand fabrication protocols. We provide a package of the bioprinter as well as the cells and media for printing of your own human skin cell models.
So when you sell these, they come in like plates with multiple wells, or how what's
Yeah,
how much
these
are they?
come in 24 well plates, but
Mhm.
you could sort of customize, you know, at the end of the day, how many walls you really want filled out of those 24 walls, or how many plates of full 24 walls you would want.
Okay.
And we would normally prepare these a few days before the terminal differentiation stage that we assess for our particular skin products. And then once you receive them, you would need to culture them in your incubation chamber, acclimatize the tissues a bit before downstream applications.
So how much does it cost with 24 well plate with in equivalence?
So without all the extraneous cost of like shipping and all that, just the well plate itself is about 400.
Mhm.
But we can give more accurate prices by official like quotations,
Yeah,
but that's
No,
about
I'm just
the price.
no, I'm just trying to get some
Idea.
general idea.
Yeah,
And but
it's really interesting. Over the last, like I think just 5 years ago, there were only a few companies sort of delivering these sort of products and services in the human skin equivalent market. But just coming from a commercial point of view, there have been companies now trying to employ novel technologies like the bioprinting to create these tissues on demand. And the products and service options have definitely expanded just over the last 3 years. And so you'll be able to find actually a lot of publications as well about how these printed tissues have already been used in your particular research areas. And if you have any questions, I can actually find those pubs for you to take a look at how they're being used. But I think the key factors would come in here about whether these tissues have been manually created or have they been printed, and what types of cells have they been used. Are these like neodermal fibroblasts, or are they making use of other types of cells representing different aging models or different, even different ethnic group models? There's also the factor of what type of ECM sort of biomaterials were used in culturing these cells. Is it just in some form of collagen? Although type 1 collagen is the most common type found, or does it include other whole ECM supplements into this environment? Like all of these things you could consider in making choice of your models.
Okay, well, thank you very much for taking the time and speaking to us. It's very interesting.
Yeah, thank you.
Thank you for the opportunity. It's our pleasure.